Biomolecules for class 12. Differentiate between pattern questions. Differences between alpha and beta glucose. Alpha glucose has a hydroxyl group of 1 and 4 position on the same side. Beta glucose has hydroxyl groups of 1 and 4 position on the opposite sides. Melting point of alpha glucose is 419 Kelvin. Melting point of beta glucose is 423 Kelvin. Alpha glucose is obtained by crystallization from concentrated solution of glucose at 303 Kelvin. Beta glucose is obtained by crystallization from hot and saturated aqueous solution at 371 Kelvin. And you can draw the diagram. So in the diagram you can see that in alpha glucose 1 and 4 OH on the same side. In the diagram you notice that 1 and 4 position OH is on the opposite side. Second question, differences between starch and cellulose. Starch is made up of alpha glucose molecules. Cellulose is made up of beta glucose molecules. Starch contains alpha 1,4 and 1,6 glycosidic linkage, whereas cellulose contains beta 1,6 glycosidic linkage. Starch dissolves in warm water. Cellulose does not dissolve in water. Starch mainly of two types of starch, namely amylose and amylopectin. Differences between amylose and amylopectin. Amylose is a straight chain polymer of D-glucose units. Amylopectin is a branched chain polymer of D-glucose units. Amylose content is in starch is about 20%. Amylopectin content in starch is about 80%. Amylose is a straight chain structure. Amylopectin is a branched structure. Amylose has alpha 1,4 glycosidic linkage. Amylopectin has alpha 1,4 glycosidic linkage and alpha 1,6 glycosidic linkage. Amylose is less soluble in water. Amylopectin is more soluble in water. Next, differences between essential amino acid and non-essential amino acids. Amino acids which the body cannot synthesize are called essential amino acid. Example, valine, leucine, etc. Therefore, they must be supplied in diet. Non-essential amino acids. Amino acids which the body can synthesize are called non-essential amino acids. Therefore, they may or may not be present in the diet. Example, glycine, alanine. Differences between globular protein and fibrous protein. Globular protein have almost spherical shape due to folding of the polypeptide chain. Fibrous protein. Polypeptide chains of fibrous protein consist of thread-like molecule which tend to lie side by side to form fibers. Globular proteins are soluble in water, whereas fibrous proteins are insoluble in water. Globular proteins are sensitive to small changes of temperature and pH. Therefore, they undergo denaturation on heating or on treatment with acids or bases. Fibrous protein are stable to moderate changes of temperature and pH. Globular protein possesses biological activities. That's why they can act as an enzyme. Fibrous protein do not have any biological activity but serve as a chief structural material of animal tissues. Example for globular protein, maltase, invertase, hormone, insulin, antibodies, transport agents, that is hemoglobin, etc. Example for fibrous protein, keratin in skin, hair, nails and wool, collagen in tendons, fibrin in silk, etc. Differences between DNA and RNA. DNA is a double strand or double helical nucleic acid. RNA is a single strand or single helical nucleic acid. DNA contains deoxyribose sugar. RNA contains ribose sugar. In DNA, the base thymine is present as a nitrogenous base along with adenine, guanine, cytosine. But in RNA, instead of thymine, uracil is present. DNA is responsible for genetic and hereditary material. RNA is involved in the synthesis of protein. DNA present in the nucleus of the cell. RNA present in both nucleus and cytoplasm. Thank you children. Keep watching, like, share and subscribe.